Okay, so good morning, everybody. Thank you for being here. Thank you for the invitation to this very nice place. I'll be happy just to tell you about some of our research lines. So it's related with viruses in general, and some of the areas that we have been investigating recently is the case of multipartite viruses, as you will see. So use evolutionary strategies for adaptation that are somehow not intuitively, say, very efficient, one would say, but they manage to do. Okay, so I have an introduction related to what viruses are. I should presume that most of you know the basics of what a virus is, but I would like to highlight some of their special features, right? So, you know, they are essentially small, typically small, but the largest viruses are known that, essentially reach the size of a small bacteria. So there is an incredible range of sizes in viruses, right? They span regarding genome sizes, regarding the sizes of their particles. They span a range of sizes that is much larger than those spread by, for instance, bacteria or other microorganisms. These are differences in different viruses. They have, so the genomes can be DNA or RNA. They have a protein capsid. Usually they are enveloped, this genetic material inside this capsid. And they might have a lipidic envelope. That's not required. Can you hear me? Because I tend to move very much. But I should speak to the microphone for the recording.
We hear well.
Oh, you hear well. OK. So, good. So what they have in common is that they are obligate intracellular parasites. So in order to replicate and to reproduce, they have to infect cells. And this is the only thing that all the entities that we call viruses have in common, because they have many, many differences, actually. We try to view them as a whole, as a unique system, but actually they are very different in the, so in genome, in the strategies they use, and also in the origin they have, as we will see. So this is not working. So this I have already said. This is the first virus that was described. That's tobacco mosaic virus. It's a very small one, and well, this is a historical curiosity. So here you have some of the aspects, so how they look like, and they might be very different, also in size, I have already said, but also in appearance, right? These are science fiction spacecrafts, sort of. This is a T4 bacteriophage. This is a very small one. They have the genetic material here inside the head, and they used to sit on top of Escherichia coli, for instance, and they push the genetic material inside through this conduct inside the cell. Ebola virus is a very large one regarding the length, and well, you see all different morphologies they might have. The classical classification of viruses has been made according to the kind of genomes they have. Either they have DNA or RNA genomes, as you see here, but they can have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA of positive polarity, of negative polarity. They can have retrotranscribed into the genome of the host, and also doing it with RNA or with DNA. And at the end, so they, of course, finish by generating a messenger RNA that generates the proteins necessary for the virus to complete the replication cycle. So you see, so many viruses have been classified inside the same group just because they have the same genomes. But then they use very different strategies regarding their evolution, or how they infect their host or propagate, right? So this is, say, valid classification, but we are much more interested in how viruses then cope with the varying environments, with the different hosts, and with the many difficulties they might encounter along their cycles. Okay, so something that you may have heard about is that viruses are nowhere in the tree of life. So this is a very interesting subject. So many people are interested in knowing where viruses come from and attending to their genomes, you might say, well, DNA viruses might be post-cellular, because if you don't have DNA, you cannot have maybe DNA-based viruses. And RNA viruses maybe are remnants of an ancient RNA world, where RNA was already able to replicate. The point is that when you look at the current tree of life, you cannot put viruses anywhere. There are some analogies, some homologies of viral proteins with proteins that are found in cellular organisms, but it's not possible to put them in a single place or to establish a history of their origin. In any way are they, for instance, monophyletic. So, as I was saying, there is this question of where do viruses come from, and if you follow the origin, so say the transition from inorganic material to complex cellular life, there might have been several places where viruses may have appeared. I mean, from the view of a physicist, I come from physics, and it's quite general that when you have a system that works, a metabolism, or something that is able to be self-sustained, parasites are something that pop out, even if you don't want them. So they are quite inherent to any working system. So I would say that any kind of parasite and a form of virus, and this is not my view, so many other people believe that this might be the case, so might have appeared as soon as you have a system that works. This might have been an RNA world, so an RNA protein world comes later, maybe comes later, and then, so you have different stages, and in any of those places you could have had parasites of a certain kind. Another interesting point is that, as I was saying, viruses are very different in nature, although we call all of them viruses, and there are also some forms that are not as clear as genetic material encapsulated in a regular capsid that then infects a host. We have other types of parasitism as well. For instance, we have these nanoviruses that do not have capsids. They have naked genomes, and they transmit vertically. We have viroids, which are a class of also naked RNA that just self-replicates, does not codify for proteins, and only infects plants. Right? They are very short. Essentially, they are circular RNA strands of about 200 to 400 nucleotides. We have also plasmids that you can also understand as a sort of parasitism, and also able to move very easily horizontally between organisms. You have introns, you have mobile elements in the genome, so you have all sorts of solutions, say, for elements that are able to just maintain their replication and are not viruses, but maybe there is a continuum. This is just to say that there are many open questions in all these areas regarding the appearance, the origin, the phylogeny, if you wish, and the evolution of viruses and related parasitic forms. Or not related, but parasitic forms. Okay, so yeah, this is the take-home message. They are inherent to life. Okay. One of the very interesting features I find of viruses is their very high modularity, probably. This is something that is under investigation also, because it's very difficult, for instance, if you're familiar with trying to see what are the pathways followed by genomes that recombine. This is already very difficult, because you can have cuts at different points, you have mixing of genomes, and even within a single species, this might be an enormous task to find out where each segment comes from. So you can imagine with viruses where the different pieces of their genomes might have come from different origins. This is a quite striking example, I think, of what viruses can do. These are nanoviruses. They have multipartite genomes. This means that the genome is not only fragmented in pieces, but those pieces are encapsulated in separated virions, in separated particles, viral particles. So in principle, they need to co-infect altogether a single cell to be able to successfully complete the replication cycle. When you look at the different pieces of genomes, that form each of these particles, you find homologies, you find some particles that are related to each other. Some of them have known functions, some of them have unknown functions, and some of them, when in vitro experiments are carried out, are dispensable, so are not essential, but they are somehow maintained in nature. So here you see that there are many questions emerging. So are those homologous pieces, for instance, the result of a duplication of the viral genome, and then one of the segments was co-opted for a different function? Or the ones that are different from those ones, are there really do they have a common origin with the other particles, or were they captured from fragments that were outside, from other viruses, or even from hosts or from bacteria? So these are all open questions, and actually when you see the picture as a whole, you imagine more a sort of a bag of genome, of solutions, of genes that viruses can use, and that are, through history, very often mixed and reused and shared between different viral families. This is probably one of the reasons that it's so difficult to reconstruct the evolutionary origin of different viral families. This is another virus in the same family, right? And this one has only 6 pieces. Okay. So, very nice example of how this modularity may have worked and is clearly seen in a virus is this mimivirus, where an analysis of the genome has shown that it has genomes from eukarya, the red part, bacteria, from other viruses, from archaea, little part, and some genes of unknown origin. So this is a very extreme and nice example of mosaicism in the genomes of viruses. Okay, so summarizing this introduction, so viruses cannot or are very difficult to be included in the tree of life. Let me just say a few words relating to that. It seems that some people also maintain that there might be some core genes in the virus that go far in the past, so farther than the phylogeny of those genomes might show. Right, so for instance, I'll show you later some history of influenza. You can go back 1 century, something like that. But it's very difficult to go much farther because you don't have information on the one hand, and then you very, very rapidly you see a divergence between the different strains. But there are some core genes that seem to push you so way far in this tree. These are genes, however. We shouldn't forget that genes might have just transmitted horizontally, so they do have a history. This does not mean necessarily that this is the history of a virus that currently is carrying that gene. Right. Okay. So they might have originated multiple times for those same reasons. And they have in common that they replicate, evolve, and adapt, and they are wonderful also model systems to study evolution. Right, because in the time of a PhD thesis, you can have nice, beautiful results of adaptation, evolution, changes in the genome, and evolutionary transitions. And I will show you one case of those. Okay, so when we think of viral diversity and all the ways they mutate and all this fragmentation of the genome and the different strategies, the different kinds of genomes, what should a model include? This is what we do in our group. We do models for viral evolution, usually inspired in experimental results. And there are many different aspects that at least you should keep in mind. It doesn't matter much if then when you try to reproduce a precise experimental result, you include them or not, because usually you cannot put all of them together. But it's important to remember that other features might be relevant or might be playing a role in that case. So for instance, we have many neutral or quasi-neutral mutations in viruses. Not in those with very small genomes, but as long as the genome is a bit larger, you very fast have these compensatory mutations and so on. So you have some diffusion in the space of genomes that is particularly acute in the case of RNA viruses that mutate very fast. You have, of course, certain fitness landscapes that people are beginning to probe, to investigate now with deep sequencing techniques. We are learning much more about that. There is a question that we don't know much about, which is the genotype-phenotype map, which is relevant not only for viruses, but in general for any model or understanding of how changes at the molecular level impact what organisms look like and what are where natural selection acts upon. Right. Then we have variable environments. This is essential for viruses. They usually have a harsh life. They go from host to host, experience population bottlenecks, are subjected to the effect of immune systems. For viruses, this is particularly difficult. When they replicate, they generate also defective forms. They generate pieces of the genome, and then they have all sorts of behavior, of dynamical behavior. This does make us think of game theory, of complementing genomes, of defective genomes, interfering forms, and so on. Right. This leads also to some phenomenology which is different from what we currently observe in cellular organisms. For instance, if you generate too many defective genomes, you might have go extinct, and we have seen this in the lab, and with certain models we have described it also, due to an excess of these defective interfering particles. Right. And then it can be also used for our quotations benefit. If we increase artificially the mutation rates, we can push viruses beyond the threshold of sustainability. They can lose their genetic information, and maybe we can induce extinction. Right. So I will just give you an example in the time that I have. I think it's about 20 minutes. Yeah. So of a case where we have investigated these multipartite forms, and we have included only 2 elements of the many that I was mentioning regarding models. This is the complementation between segmented genomes, fragmented genomes, and what happens in a variable environment. One example that I didn't want to go into the case that I'm going to treat without mentioning, because you have heard about it certainly, is that of influenza virus. Influenza has a fragmented genome, has 8 pieces, 8 chromosomes, right? But they are all inside one single viral particle, right? So this case is different from the one that I'm going to talk about in a minute. Yeah. And what it permits to influenza is to very easily share or exchange those segments with other genomes. So you don't need a bona fide recombination, but you can just, if you have 2 influenza particles infecting one same cell, you just can shuffle the segments between the 2 particles. This is the risks so you remember some time ago, this avian influenza, right? So if you have co-infection of one host, of the same host, with 2 types of influenza that have the same structure, then for instance, one of the segments causes a high virulence, and another one is able to permit the transmission from host to host in an easy way, and you combine both, then you can have a pandemia due to this reason, to this combination. And this is the danger. So I was saying previously, here you have a phylogeny also, because we know it's influenza, because it maintains, in a sense, the phenotype. This is the symptoms. This is not influenza, what I have today. It's a rhinovirus, but it's also, it's there. And you can see the number of nucleotide differences it has accumulated from 1965 to 2000, approximately. It's nice also because the colors, the change in the colors means an antigenic change. So this means that when you change the color, you fail to be recognized by the cellular receptors that you had in the previous season, and then you have essentially a new form that needs to compete again, so the immune system has to fight against it again, right? So you have, say, these might be changes in the phenotype while you have continuous changes in the genotype, right? This continuous accumulation of mutations. So you see, this is so fast that it's very difficult, makes it very difficult to go very far into the past. So no doubt that once you have the ecological niche, say humans that have noses and mucus inside, and so you will have some parasite that takes advantage of that ecological niche, right? But it might have been also a different form. So phylogeny is really a difficult issue in viruses. There is a lack of continuity probably when you go to the past. Okay, now let's go to the case of bipartite viruses. Here we have fragmented genomes, but as I was saying, those genomes are encapsulated in different particles. This means that if I have a cell and I want to successfully infect that cell, I need both segments to go inside the cell. So I need 2 viral particles attacking and putting the genetic material inside the cell. It's quite interesting that those viruses, multipartite viruses, are only found in plants. They only infect plants. This is probably due, but this is something that one can discuss because things in evolution are never as easy as they seem. This might be due to the kind of tissues plants have and to the fact that they promote an increase in density because of this structure, of the spatial structure of the plant. Right. Typically, those viruses also are transmitted through vectors, through insects. So insects usually take a large amount of virus, go to another plant, and put also a large amount of virus. In this sense, if you avoid population bottlenecks, it's easier that you maintain a high multiplicity of infection, and then that you successfully can maintain the infection, even with a genome that requires co-infection by 2 different viral particles. One of the hypotheses for the origin of those viruses is maybe that they made a transition which is inverse to many of the reverse to many of the transitions we have seen. So typically, we know of going from isolated elements to elements that cooperate. This is a transition from competitors to cooperators. So you have, in the origin of life, we may have had those molecules replicating and taking products of other molecules. So, well, the solution to construct systems that work is to have those hypercycles or those, say, cycles involving many different catalysts. And at the end, what you do is you go to something that is inside a compartment and then is forced to replicate all together. And this is a benefit for all of them, in a sense. And you avoid so the isolation of elements and, in a sense, control competition. We have seen this also from when we go from unicellular organisms to multicellular organisms. We have seen this in insects, going from solitary individuals to colonies of insects, and also at the molecular level, so the transition to chromosomes. And in viruses, in those multipartite viruses, one could think that this transition has occurred in the opposite way, that you may have had some complete genome here, and you have made a transition to dividing this genome and having 2 separate parts that in principle complement, but who knows if at some point they might compete. Anyway, this was just to put it in an evolutionary perspective. And in this sense, so it might be that when you have this wild-type full genome, say, for instance, you have a very simplified genome where you have a replicase gene and a gene codifying for the protein of the capsid of the virus. Typically, they have always these 2 proteins. You have this independent replication if you have a full genome, so they replicate both. And very typically, through replication errors, you have deletions of parts of the genome. And occasionally, it might have that you just, by chance, separate the 2 genes that form the genome of the virus. And then you might experience a transition to complementation instead of having the full genome together. Okay, so in this case, you have 1 segment per capsid, and there is a drawback, of course, which is that if you infect separately, and you don't find the complementing segment, you are not able to proceed with the infection, and you need this complementation. Okay, let me see. Well, okay. Some colleagues of mine, Esteban Domingo at the Center for Molecular Biology in Madrid, decided to undertake this experiment and see if viruses, under conditions of high multiplicity of infection, could really experience this evolutionary transition from a complete genome to a fragmented genome with segments that complement each other. And of course, the question behind was, what is, if any, the advantage of fragmenting the genome? Because it seems quite counterintuitive that you might have any advantage at all. It seems that a complete genome might be always better, right? They adapted to culture foot and mouth disease virus, which is an animal virus, not a plant virus. It's an animal virus. And PhD thesis, I think there were 2 or 3 of them in that. They start to plague the virus, to infect cells at high multiplicity of infection, taking samples of what resulted from there, and continuing this procedure with the hope of seeing the emergence of those 2 complementing fragments. Well, it seems quite amazing. I feel still amazed by the experiment. But after 260 passages, it turned out that they found 2 defective complementary forms. So they were unable to replicate without the help of the other part. They were encapsulated in different particles, and they could compete against the wild type. So this is just an example of how they separated. This was an artificial separation at the beginning. The 2 complementary forms. This is so they are effective at causing lysis of the cells. They caused smaller plaques, if you want. And this is the case of the wild type. Okay, so this worked. And then, so the question was, okay, are we going to see that the complementary forms are better than the wild type? In which conditions? We know that if you need complementation, this requires high multiplicity of infection. On the other hand, there were in the literature, you can find at least 2 situations where maybe you might be at an advantage if you have a shorter size in the genome. Maybe you can replicate faster. You can replicate the 2 segments in parallel faster than just having to replicate a longer wild type. And maybe you might have a higher copying fidelity in the sense that you might have sort of sex working in those viruses and just so get rid of deleterious mutation in an easier way. So these were the proposals in the literature. They tried to see if any of those was working there, and they were not there. So there is no faster replication, so significantly. There is no higher copying fidelity. And it turned out that the advantage, and this took another 7 years, I think, to find out, because the first paper we published in 2004 with the transition, and the paper with the role of increased stability of the viral particle is 2011, I think. So it took a good number of years to find out. And it is because shorter genomes in very compact viral particles exert less pressure on the capsid, and their average lifetimes are longer. And this made the difference in this case. Then we had the model to try to describe the whole viral cycle. We have the 3 types that I have described, the wild type and the 2 segments. We infect cells at a given multiplicity of infection, taking an average multiplicity of infection, this M here. And so just infecting randomly. We replicate the viruses inside. That depends on the intracell composition, if I have or not complementation. The wild type can replicate on its own. And then we have a differential degradation, which is reflecting this idea, this observation, that so genomes with smaller genomes have longer average lifetimes when they are in the virion form, right? And we start a new infection. So we continue this cycle. And these are numerical results now of what happens there. We have 2 different parameters, which are the multiplicity of infection and the degradation of the complementary forms with respect to the wild type. And what we see is that we have a transition from coexistence of the three types to extinction of the wild type, where when we cross a line where the advantage of complementation is sufficiently high, either regarding significant degradation of the wild type with respect to the other forms, or either because we have a sufficiently high multiplicity of infection. And then the wild type can disappear. What is nice is that here we have the results of experiments. We could infer the value of the two parameters relevant for the model, and we saw that there were different results depending on the phase where the dynamics of the systems were taking place. So essentially, these are the evolutionary outcomes. If we plot here, so the densities at the stationary state of each of the types, wild type and the two complementary forms, we see that by symmetry, we always have the same density of the two complementary forms, so we are along this line. And in the case of coexistence, we have so a varying value of the stationary density of the wild type, as it is represented here, and the same density of the two complementary segments. Right? So these are the two phases that we see. Okay, this is just to, I could have put more equations. Actually, we can solve exactly the transition line, which was a nice result also. And here, this dotted line, this dashed line, is the exact result of the calculation. And these are the numerical results. So as you can see, we can capture very well where the transition occurs. Right. Okay. Also, something that we did was to try to figure out where the two other hypotheses in the literature regarding where were the advantages of these shorter genomes were. And we got just to say that we can put in correspondence the different parameters, and the phenomenology would be exactly the same. So if it replicates faster, or if it has a higher fidelity of copy, then we can just find the appropriate parameters that would describe the other situation, and we would have, again, the two phases where we have either coexistence of the three types or extinction of the wild type. Yeah, I should finish. Okay. The problem comes when we try to apply the same reasoning to a situation where we have more than two fragments. Because the multiplicity of infection, just let me think of this line here. Just look at this yellow line. This means, what is the multiplicity of infection that I need for the bipartite form to be fixed instead of the wild type if I have two segments? If I have three segments, my multiplicity of infection raises very much. If I have 4, I go to 100. If I have 5, and just, okay, this is 6, yeah, right? For 6 segments, I need about 1,000 particles infecting a virus for this procedure to work. And this is nonsense. Though in nature, we see fragmented forms, or multipartite forms, with up to 11 different particles. But of course, this cannot be the way of generating those viruses. That's an open question, again. So what is then the mechanism that permits that? And this is not yet answered. So we are working on that, trying to see if there might be other strategies that allow this, this solution, and is not due to the need to maintain this very high multiplicity of infection. There are other questions related that emerged from the model. And one of those, for instance, is what is the density of fragments that I should observe when I have these multipartite viruses? In our case, in the case of the model, due to symmetry, you should have equal densities. But when you go out to the field and measure the abundance of different segments in viruses, in this case with 8 different segments, you see that the relative frequency varies very much from segment to segment. We don't know precisely what we are seeing here. It might be a matter of regulation, it might be a matter of proteins that are more needed than others, and the virus somehow manages to have this unequal distribution of the segments. But as you can imagine, this requires then, if I have fragments that are in a very low frequency and they are maintained, then I cannot think that a higher multiplicity of infection is going to solve for the maintenance of this segment. So I have to go to resort to another solution.
There's no chance that by mutation you can shift from one genome to another?
No, not in those cases. I mean, this might happen, but at the time scale and at an evolutionary time scale, this is much larger than what you see here. So this is just infection from plant to plant. And you label your segments so you know they are there. It's not that you are generating the other. So
I just meant that the R segments cannot become S segments by mutation.
Not in the time scale of the experiment. I mean, yeah, in 100 years maybe, or yeah, but no, not at that time scale. So there should be something else that permits that they are maintained in the system. This is another example with another RNA virus, alfalfa mosaic virus, and you see again the very different frequencies of segments. These groups are working on trying to figure out what's going there. This is a group from here, from France, from Stéphane Blanc, and this is a Spanish group, Santiago Elena. Okay, so let me just summarize. As you can see, I have said very few about symptoms of viruses or viruses as elements that cause disease, because I think that their interest and probably the amount of viruses out there that we don't know about, but that have important roles also in evolution, is much larger than those viruses that cause our symptoms or cause disease. They may have originated multiple times, are so adapt very rapidly, have discovered many different, or have discovered in a non-purpose sense, many different strategies, among them the one that I have been talking about today, which is this fragmentation of the genome. They are really a window to peep into how evolution at the molecular level can work and what are the procedures that it might use. And we can learn a lot, I guess, from viruses of mechanisms of evolution and adaptation. This is something that I didn't talk about today, but there are also analogies with early replicators that might also shed some light on how the first replicators on Earth might have evolved. Okay, so just my acknowledgements to my colleagues, especially, very especially to the lab of Esteban Domingo, who performed many of the experiments where I have been working, doing models, and Jaime Iranzo, now at the US, who did part of his PhD thesis were the results that I have shown today. Thank you very much.